is Adebola Oluyede. I'm a senior at North Point High School, and along with my mentor, we did an investigation of neural information and a comparison of CRISPR-edited microglial cells to disease cells in a neurodegenerative disorder called Friedrich's ataxia. Oops. Okay, so what is Friedrich ataxia? It's a neurodegenerative autosomal recessive inherited disease that results in damage of the central and peripheral nervous systems over time. Such damage results in difficulty moving, walking, a lot of tiredness, loss of reflexes, slurred speech, vision loss, and patients also tend to experience a lot of cardiovascular and heart issues as well. But what causes FRDA, also known as Friedrich ataxia? So it is caused by a GAA trinucleotide repeat in the frataxin gene located on chromosome 9. Within a healthy person, there are around 5 to 30 GAA repeats within the frataxin gene. But however, in FRDA, there are hundreds of copies of this repeat, which affects the transcription of the protein known as frataxin. If you look at it in figure 1, you can see the normal state, and it has about, it looks like 20 repeats, but there's about like 500 of these, and it results in less frataxin being made, thus resulting in a deficiency. So what is frataxin? So it's a small, highly reactive, conserved mitochondrial protein responsible for a variety of functions within a cell, but it mainly relates to cellular regulation of iron homeostasis. And deficiencies in this protein caused by FRDA have been known to cause mitochondrial iron overload, thus resulting in oxidative damage in mitochondrial and nuclear DNA. So the cell of interest, microglia. So they are the brain's resident macrophages, and they play a variety of roles within the brain, ranging from breaking down of products, to supporting myelination, to neurogenesis, to cell death or cell survival, or stimulating synaptic formation. So protein aggregation and neural damage can cause the activation of disease-associated microglia through damage-associated molecular patterns. So these disease-associated microglia can further neurodegeneration that is already happening due to FRDA by increasing oxidative stress through the production of reactive oxygen species. So we're here to move on to our proposed solution, CRISPR. So what is CRISPR? It's a tool used for editing genomes, allowing researchers to go ahead and change gene sequences and modify gene function. So if we segue into that, what we're hoping to do is to use CRISPR. Oops. My bad. Okay, so what we're hoping to do is to investigate whether the use of CRISPR gene editing software can be used to correct the GAA mutation that we saw earlier with way too many repeats that results in FRDA. And we're wondering if we can decrease the activation inflammation in microglial cells. The way that we're going to quantify this activation is through immunofluorescence, meaning that patient-derived cells are going to be stained with different dyes, photographed, then further analyzed using image analyzing software. We were then able to make a comparison between the edited patient-derived cells and the unedited FRDA cells. What we hypothesized is that through CRISPR editing to correct this mutation, we will see less activation in the lysosomes and mitochondria of microglia cells, thus suggesting less activation overall. So how did we do this? So FRDA patient lymphoblasts were edited using CRISPR to remove the GAA trinucleotide repeat expansion. And you can see that in figure 2. And once we did this, once we separated the edited as in corrected and the FRDA as in diseased, we were then able to reprogram them into stem cells. And upon doing this, we go ahead and differentiate them into microglia that we can use to assess their phenotype and function. So how did we go about assessing these microglia? So like I said earlier, we used immunofluorescence. We use this to investigate potentially linked phenotype in microglia and FRDA. Images were stained using CD68 antibodies, which stain the lysosome of the cell, and mitosocs, which stains mitochondria, and it investigates superoxide levels since frataxin is a mitochondrial protein. So upon collecting the stained samples, the software image J was utilized to split the overlaid images into different channels, and we saw these in grayscale. And from this, we derived cell count, the area, and the mean grayscale intensity of each sample. So the mean grayscale is important because it shows us the amount of intensity that is given off on the image, which shows how activated a cell is or isn't. So through the things that we got from image J, we were able to calculate the mean grayscale intensity per cell by dividing our mean grayscale value by the number of cells in any given image. So remember that higher mean grayscale intensities could potentially mean that the mitochondria and lysosomes are more activated. And the higher number of activated lysosomes within a cell, the more inflamed and activated the cell is. So these are the images that we got. At the top here, we have the CD68 lysosomal images at 20 magnification. You can see at a first glance that the FRDA images look a lot more illuminated in comparison to the edited images. You can see a lot more cells, you can see a lot more red being given off. And we had 6 CD68 images, 6 CD68 stained images. And we had 6 mitochondrial images. So we went ahead and analyzed each images for their mean grayscale value. And that is concluded in the table. So we went ahead and took the average of the mean grayscale intensity for each image and found the average per cell overall of all these CD68 samples that we had. So, and we did the same for mitosocs. So at a first glance, you can see that the edited images have a lower average grayscale per cell than the FRDA images, which suggests that they are less activated in comparison to the FRDA images. And this is a graphical representation of the CD68 tracker and the mitosocs tracker. We can see clearly that the edited bars are lower than that of the FRDA bars. So what does this mean? So analysis of this image reveals that CRISPR-edited microglial cells had less mitosocs and CD68 activation in comparison to FRDA cells. Figures 5 and 6, the graphs that I just showed before, show the grace intensity per cell. And in both figures, the FRDA bar is higher than that of the edited and corrected bar. So in this study, we used CD68 as mitosocs as indicators of activation and inflammation within a cell. And based upon the data that we saw in tables 1, 2, figures 5 and 6, it's clear that CRISPR edited cells experience less inflammation than FRDA cells. So as of now, it's been around 165 years since, 160, years since Friedrich ataxia was discovered, but there still is no cure and no effective treatments as of now. Through the use of gene therapy, we hope that we can go ahead and correct the main cause of this, the genetic cause of this disorder through the editing of the cell and to correct the repeat. So while we were able to compare inflammation between diseased and edited cells, we didn't test whether the frataxin protein levels were restored to normal after CRISPR editing, or whether there was a decrease in progression of degeneration in patients. But these limitations show us that the path to creating a treatment for Friedrich ataxia is long, but not impossible. This study serves as a step in the right direction, showing that use of CRISPR gene editing software does make a difference in the activation of edited microglia versus diseased microglia. The next step in development of this treatment is looking at the effects of the GAA trinucleotide mutation on frataxin production levels of reactive oxygen species and oxidative stress within microglia, and even expanding such analysis to other types of cells in the brain that are also affected by FRDA. So in conclusion, the purpose of comparison between the diseased affected microglia and CRISPR gene edited microglia was fulfilled. Collection, creation, staining, analysis, An analysis of microglia revealed that CRISPR-edited microglia showed less average grayscale values when stained with both CD68 antibodies and mitochondrial superoxide dye than their diseased microglia counterparts. Such suggests that the edited cells were less activated and less inflamed than disease cells, meaning that correcting the frataxin gene did make a difference in terms of inflammation. Based on these results, the next step would be to perhaps apply the same correction to other FRDA-affected cells, see if similar results occur. But regardless, the prospect of gene correction to treat diseases like FRDA creates hope not only for FRDA patients, but for the many, many patients who suffer from other genetic or neurodegenerative conditions. So these are the references that I used. And I also just wanted to say that I really, really enjoyed my time here. I really enjoyed a lot of the research that I went into. Me personally, I was really interested in doing a project on neuroscience, and that is exactly what I got to do. I really thank my mentor, Ms. Avalon Johnson, for all the help that she gave me and breaking down this really complex disease and being able to present this in a way that people can understand. So yeah, I really appreciate this opportunity, and thank you all for listening to my presentation. So thank you.